Welcome back to Sciency Stuff with Julia and Janae featuring Connie and Haas. Today we will be talking about the wonderful and sciencey process of protein synthesis, or the process where amino acids are arranged with help from ribosomal RNA, transfer RNA, and messenger RNA. Before we talk about the process of protein synthesis, we must understand the structure of DNA. DNA is a double helix and consists of two strands containing deoxyribose sugars and phosphate groups running parallel to each other in opposite directions, aka it is anti-parallel. One DNA strand starts at the top with the first phosphate bonding to the fifth carbon of the sugar and ends with the third carbon, and it is called the 5' strand, while the other strand is reversed and starts with the third carbon and ends with a phosphate bonded to the fifth carbon of the sugar and is called the 3' strand. Each step of the ladder is made up of a pair of nitrogenous bases bonded by a hydrogen bond, which is a bond between an electropositive hydrogen in one molecule and an electronegative atom of oxygen, nitrogen, or fluorine in another molecule. In DNA, the hydrogen in one nitrogenous base, NH3, forms a hydrogen bond with the nitrogen in the nitrogenous base it is paired with. Guanine pairs with cytosine, and thymine pairs with adenine. Each set of sugar, phosphate group, or nitrogenous base is called a nucleotide.
Now let's talk about transcription, the first step of protein synthesis. Transcription occurs in the nucleus. First, RNA polymerase binds to specific regions of DNA called promoters, which tell it where to start reading. Then it unwinds the DNA it is instructed to and copies the template strand by creating an mRNA sequence of nitrogenous bases that are complementary to the DNA's nitrogenous bases. Eventually, the mRNA stops at a termination sequence. Here, the DNA code TAC, GCC, TGA, CTG, ATT is transcribed into the mRNA sequence AUG, CGG, ACU, GAC, UAA. In eukaryotes, pre-mRNA has introns, which are sections of mRNA that do not code for amino acids, and exons, sections that do code for amino acids. During its last moments in the nucleus, the mRNA goes through splicing, and introns are cut out and the exons are joined together. Then it is known as mature mRNA. After, a methylated G-cap and a poly-A tail are put on its ends.
The second part of protein synthesis is translation, the process of creating a protein by matching specific amino acids to the mRNA sequence. First, mRNA leaves the nucleus and goes to a ribosome, which is where translation occurs, and then the ribosome looks for the start codon AUG, which codes for met or methionine. Afterwards, the ribosome reads the mRNA strand in triplets called codons. tRNA's anticodons match with them and bring corresponding amino acids to attach to the growing polypeptide chain. At the end, when the ribosome reads a stop codon, UAA, UAG, or UGA, it stops translating the mRNA. Here, the mRNA sequence is translated into the amino acid sequence MET, ARG, THR, ASP, stop. Now let's talk about ribosomes. Ribosomes are made up of rRNA and proteins and have three binding sites. The A site, where incoming tRNAs with amino acids land. The P site, where amino acids are joined by peptide bonds and the polypeptide chain grows. And lastly, the E site, where the tRNA exits the ribosome.
Let's get a close-up of the polypeptide chain. Peptide bonds are covalent bonds formed between two amino acids. They bond the amino group of each amino acid to the carboxyl group of the next and are located between the amino acids of proteins.
Now Anthony is going to wrap a summary to go over everything.
Yo, it's your boy Haas, and it's time about to recap. The first thing to understand is that DNA is a double helix with anti-parallel strands that are 3' and 5'. They contain deoxyribose sugars and phosphate. As the strands put together side by side, forming the ladder as the DNA pattern binds in order to produce nucleotides. The first step of protein synthesis starts off with transcription deep inside the nucleus. The RNA polymerase finds the promoter, then binds it in order to rewind the DNA and copy the type of strand. So it's complementary to the DNA nitrogen space at the aggregation and termination. The process is ready for the next phase. But now comes the introns and extrons from eukaryotes. When they're together, the messenger RNA is immature. But when the introns are sparse, it goes thin as mercury for sure. The last part of protein synthesis is translation, so the amino acids take on a new destination to the ribosome, so they can be seen as a brand new protein.